blood groups, red blood cells have certain proteins called antigens on their plasma membranes. These antigens. Now, these antigens stimulate the immune system to produce specific antibodies. So, an antigen is any substance, usually protein in nature, that stimulates the immune system of an organism to produce antibodies that are specific to it. Now, based on the presence of certain antigens, there are two main blood grouping systems, namely the ABO system and the Rhesus factor system. In the first system, that is the ABO system, there are two types of antigens designated A antigen and B antigen. Blood groups are determined by the types of the antigen present in an individual's red blood cell. Thus, an individual whose red blood cells have A antigen is grouped as blood group A. So, this is a red blood cell. If on the plasma membrane there is antigen A, then that blood is grouped as A, blood group A. Likewise, an individual in whose blood the red blood cells have B antigen, that blood group is B. Then there are those individuals on whose red blood cells antigens A and B are both present. So here you have antigen A, B antigen is present, so that group is designated as AB. While those individuals whose red blood cells lack neither antigen A nor B are said to belong to group O. So basically there are four blood groups under the ABO system. Blood group A that has antigen A, blood group B with antigen B, blood group AB that has both antigens A and B, and blood group O that lacks neither A antigen and B antigen. Likewise, the plasma too has antibodies. Antibodies that are present in the plasma which correspond to antigens A and B. There are two types of these antibodies designated antibody A, small letter A, and antibody B. Antibody A reacts with antigen A, while antibody B reacts with antigen B. Now, if an individual has antigen A, that is blood group A, he or she cannot have antibody A. So naturally, individuals that have blood group A have antibody B in their plasma. Likewise, those people that belong to blood group B, meaning they have antigen B on their red blood cells, have antibody A. While those that belong to blood group AB that have both antigens cannot have either antibody A or B. So they have no antibodies in their plasma. They have neither antibody A nor B. While the blood group O that has no antigens on the red blood cells, has both antibody A and antibody B in the plasma. Now, as mentioned, antibody A, for instance, reacts with the antigen A. So if red blood cells that say have antigen A comes into contact with blood or plasma that has antibody A, there's going to be clumping together, or what is known as agglutination. These antibodies will cause the clumping together of the red blood cells that have the corresponding antigen. So it is very important that antibodies and antigen do not correspond, or they do not occur together in the same circulatory system, for there is going to be an antigen antibody reaction or antibody antigen reaction resulting in agglutination. And this is what is taken into consideration during blood transfusion. Now, blood transfusion refers to the transfer of blood from a donor into the circulatory system of a recipient. A recipient will receive blood from a donor if the recipient cannot produce antibodies corresponding to the donor's antigens. That is, the transfusion is going to be compatible if the recipient's blood does not have corresponding antibodies to the donor's antigens. So during transfusion, what is taken into account? You look at the donor's antigens vis-a-vis the recipient's antibodies. Because if the donor has, say, antigen A and the recipient has antibody B, then there's going to be an agglutination. There's going to be an antibody-antigen reaction. So during transfusion, we look at the recipient's antibodies. By knowing the recipient's blood group, we can tell the antibodies present, and then we match that to the donor's blood group. If the antibody and the antigens correspond, then we avoid the transfusion and we say that two bloods are incompatible. Now in this table here summarizes the compatibility of the different types or groups of blood during transfusion. We take blood group A with antigen A, has antibody B. So if blood group A is the donor, meaning that blood group A can give to another blood group A and can give to AB. Why AB? Remember, AB does not have any antibodies, so it will not matter the type of antigen present on A. Whereas blood group A, if he is a recipient, he can receive from another blood group A and blood group O. Because blood group O does not have antibodies that will react with the antigen in his red blood cells. Blood group B has antigen B and antibody A. Therefore, if B is the recipient, he can receive from another B and O, but not A, because the antibody A will react with the donor's A antigen. Blood group B can give to another B, of course, and can also give to AB, because AB does not have antibodies to react with the B antigen. Blood group AB does not have any antibody, meaning that can receive from AB, can receive from A, can receive from B, can receive from O, because it has no antibodies to react with the donor's antigen. For this reason, blood group AB is said or is referred to as universal recipient. Individuals that have blood group AB are said to be universal recipients because they can receive blood from all the other groups since they don't have antibodies to react with the donor's antigen. Blood group O does not have any antigen but has both antibody A and B, meaning that O can receive only from O because it has the two antibodies and can only receive blood that does not have any antigen, and that is blood group O. However, blood group O can give to itself, can give to A because it has no antigens to react with the antibody B in blood group A, can also give to B since it has no antigen to react with the antibody A in blood group B, and can also give to AB since it has no antigens to react with any antibodies. But also remember, blood group AB does not have any antibodies, so it can receive from blood group O. Now, because blood group O can donate to all the others, the blood group O is said or is referred to as universal donor. It can give to all the other groups because it lacks antigens. Blood group AB is universal recipient because it lacks antibodies that will react with the donor's antigens, while blood group O is universal donor because it lacks the antigens to react with the recipient's antibodies. Now, in addition to the ABO system, there is another grouping system based on another type of antigen, that is the Rhesus factor antigen. So the red blood cells may also have another antigen on their membrane known as the Rhesus antigen. Individuals with the Rhesus antigen on the membrane of the red blood cells are said to be Rhesus positive, while those individuals whose red blood cells lack the Rhesus antigen are said to be Rhesus negative. Now, on average, about 85% of the population are Rhesus positive, and only about 15% are Rhesus negative. Now, normally, the Rhesus negative person does not have antibodies against the Rhesus antigen. Normally, the Rhesus negative person does not have antibodies against the Rhesus antigen. However, when Rhesus positive red blood cells are introduced into the circulatory system of a Rhesus negative person, then they will develop the anti-Rhesus antibody. So normally during transfusion, to prevent development or formation of anti-Rhesus antibodies, Rhesus negative persons are not transfused with Rhesus positive. So for example, if one is blood group A negative, meaning that they have antigen A, but they lack the Rhesus antigen, then they can only receive blood from A negative and O negative. Because if you transfuse this recipient with the A positive blood, then the Rhesus factor on the red blood cells will cause the formation of antibodies against the Rhesus factor, and that may lead in future to agglutination. So Rhesus negative persons receive Rhesus negative blood from corresponding groups. If it is B negative, gets B negative or O negative, but not negative recipient getting positive, Rhesus positive blood. But on the other hand, these Rhesus positive individuals can receive blood from Rhesus negative individuals without any harm. Now, when a Rhesus negative woman marries a Rhesus positive man, the woman is likely to conceive a Rhesus positive fetus. So you have the Rhesus positive father, Rhesus negative mother. This fetus here is most likely to be Rhesus positive. Now, the Rhesus positive antigen of the fetus may pass across the placenta. May pass across the placenta into the mother's bloodstream during the last month of pregnancy. The mother's bloodstream or the immune system responds by producing Rhesus antibodies. So the Rhesus antibodies are produced in response to the presence of the Rhesus positive red blood cells. Now, the Rhesus antibodies destroy some of the red blood cells of the fetus because these antibodies can pass across the placenta back into the fetal circulatory system. So, within the fetal circulatory system, the Rhesus positive red blood cells are destroyed by the anti-Rhesus antibody. Now, the firstborn child has a higher chance of survival because the destruction of the red blood cells is minimal. However, in the subsequent pregnancy, if the fetuses happen to be positive, then there's going to be massive destruction of the red blood cells, leading to serious symptoms and deformities of the baby, a condition known as hemolytic disease of the newborn, or erythroblastosis fetalis. However, to prevent erythroblastosis fetalis, the mother can be treated with anti-Rhesus globulin. Anti-Rhesus globulin, which prevents her from producing antibodies against the fetal antigens. So that during the last month, she's treated with this anti-Rhesus globulin so that the presence of the Rhesus positive red blood cells is not recognized by her immune system. So she will not develop anti-Rhesus antibodies. This will protect the red blood cells of the fetus in the present and the subsequent pregnancies from destruction. However, after birth, if the baby is still alive, the baby can be transfused with the Rhesus negative blood to remove the destroyed red blood cells and also get rid of the Rhesus antibodies in the baby system so that the circulatory system can function normally. So whenever a Rhesus positive father marries and have a child with a Rhesus negative, the high chances of getting a Rhesus positive. Now, in the first pregnancy, the baby may not suffer as much because by the time sufficient antibodies are produced, the baby will have been born. But the subsequent ones are the ones that will suffer because these antibodies remain. They linger on in the mother's circulatory system and would invade the subsequent fetal circulatory system. Should the baby be Rhesus positive, then there will be massive destruction of the red blood cells.